And our final speaker for this session is Scott Olson from the US CDC Center for Forecasting and Outbreak Analytics. Over to you. Thanks so much.
Thanks, Jillian. My name is Scott Olson. I'm at the CDC Center for Forecasting and Outbreak Analytics. Our mission at the center is to improve public health decision-making. So we're a little different maybe from other modelers in that our goal isn't necessarily the generation of knowledge. Like our primary goal is the improvement of decision-making. I'm not saying that's not anyone else's goal, but the things that I have to say here are lessons from thinking about modeling for wastewater and COVID-19 variants in the context of that kind of ideal of firmly rooted in the data that we have, but also where we want to go. So this is very much a talk for modelers about modeling. I hope that you find that interesting. Okay, so wastewater data. We've talked about this a little bit. Wastewater surveillance is a promising technology. There's a lot of reasons for that. Representative, efficient, community-wide sampling, the potential for parallel quantification of many different pathogens, either through metagenomics or through lots of parallel pathogen-specific quantification. There's a potential for early warning. As we've talked about, there's a few different kind of delays here. A delay from infection to shedding, a delay from infection to healthcare seeking, which is perhaps somewhat malleable, and then a delay between a healthcare encounter and reporting. What do we mean by reporting? In the US, at least, this can be a very complicated question because you can have a healthcare encounter at a hospital. The hospital will aggregate your data, send it to a US state that later will send it to the CDC. There can be a long delay in there. What you mean by reporting and what you mean by delay and early warning depends on that definition. Okay, so there's all this promise, but in practice, the challenge of data to action for wastewater has been tough. Integrating this kind of data stream into public health practice has not been without its bumps. And so I wanted to point out, these are just like a few representative tools that I think that have the goal of taking wastewater to action. So on the right, you see this tool called COVID Surge, which I think is an interesting example. The idea there is that if you have a sequence of, by sequence, I mean a set of samples from a single location, and you want to say, is the data that I'm getting from this location telling me that I'm about to have a COVID surge? So the analysis there is sort of a decision tree where you say, okay, is this sample high enough relative to previous ones that I think something bad is going to happen? What you see on the bottom is the old CDC's visualization tool for wastewater, where each site was analyzed separately, and there was kind of a regression that said what is the growth rate within that site. This was potentially useful for people who were working within each site, but folks who were trying to work at one level higher found it a bit difficult to work with, which inspired what you see at the top here, which is an aggregated signal across regions, and that has some temporal baseline in it. So this is kind of nice because it makes it easy to look at and say, are we at a peak or not? But as you might imagine, there's a whole lot of information that is lost in this aggregation. And so the question is like, why can't we do more with this really rich data stream? We have hundreds of sites, we have thousands of samples. Like, we should really be able to get more out of it than either, a bunch of colored dots or a few colored lines. So why is that tough? One is that wastewater data has intrinsic biological noise. I don't think there's a way that we can get around this. Different people are going to shed at different rates. There's much better data about variability in nasal shedding and fecal shedding. So I'll just point you to this example that probably peak nasal shedding for COVID-19 varies by 2 orders of magnitude probably. And even without that variability between individuals, there's just this like shot noise problem. So if the wastewater signal is the mean, the average of contributions of people in a wastewater catchment, it says infected people, it should just say individuals in a watershed. Then your noise in that signal is going to depend on prevalence and on population size. So anyone who's worked with wastewater data at small scales, it looks crazy spiky, but that's not necessarily any error in the data. That's just because you have so few people who can be having this 100-fold difference in shedding. So I don't think this is a kind of noise that is intrinsic to the data that we're not going to get around. There's other kinds of noise that we have introduced in the systems that we use, right? So, oops, I got to click forward. Got to keep track of both of those. So in the United States, at least, in the National Wastewater Surveillance System, there's a lot of variety in the reporting cadence from different sites. Some sites have pretty high sample to sample variability, others lower. There are sites that have consistently higher or lower SARS-CoV-2 measurements that come out of their wastewater. Wastewater is not a consistent thing. It can be really different from location to location. Every wastewater treatment plant in the United States is different. There's not even a single list of all the wastewater treatment plants in the United States. It's a very fragmented kind of system. Some sites report different times during the week. They have different reporting delays. Sites can drop in and out of the surveillance system, and they can use different lab measurement systems. So there's a lot of noise in here also due to all of these other pieces. Okay, so I've said why it's hard. Like, okay, what's there to do about it? One is, when possible, as Frank was saying, decrease the variability in methodology. So there is this recent National Academies report that calls for limiting and standardizing sampling methodologies, so the physical transfer of the wastewater out of the wastewater system and into the lab, and then some effort to consider standardization of wet lab methodology. And then, the dry lab stuff is even one step further if we're talking about metagenomics. So where possible, decrease variability and methodology. This just came out a couple weeks ago, which is maybe why it seems new. But where that's not possible, then I think it was Phil who said this early in the day, we should build our analytical tools that incorporate and work around these known variabilities. So I'm just pointing to some work that Dr. Caitlin Johnson at CFA worked on to build a model of wastewater and hospitalizations that can work across all the states in the United States and across all of that incredible diversity. And our hospital reporting system is changing for COVID now, but this gives you a sort of sense of how things work, is that states will report hospital admissions at the level of the entire state, and then each wastewater catchment will report at their own level. And so you need a model that can deal with that grouping and that can deal statistically for the variations between sites, both in terms of wastewater facilities, wastewater lab methods, and soon also hospital admission reporting methods, which can be different from place to place. If you're interested in what that looks like, it's being submitted to the COVID-19 Forecasting Hub, and there's a link down there that will be on a later slide too. But the overall idea is that there's a Bayesian fusion signal model that takes hospital admissions data and wastewater data in and forecasts hospital admissions out. So you have some epidemiological model on the back end, and then you have some observation processes on the front end that account for the site-to-site variability in these different places. This is the result of a lot of thinking and discussion, and I think this is the way that this data should be used when you have the clinical data to do the integration with. So if you violently disagree with this, I definitely want to talk to you at happy hour and figure out what we can do there. But also happy to point folks to this kind of work if it's going to advance your thinking. At CFA, we want to build tools that advances everyone else's work. So if you find it useful, we think that's great. We're just trying to save people time and effort where we can. Okay, and switching gears to a totally different thing, and this is mostly the result of talking to Dr. Andy McGee in my group at CFA. I just want to give him so much credit for all of this. Thinking about COVID-19 variant forecasting and the challenges that arise there and what we can learn from them, again, through this embracing of heterogeneity. So CDC produces variant nowcasts. They're called nowcasts basically to make really clear that there's a long reporting delay between the collection of the specimen and the reporting of the result. And so the nowcasting is forecasting, but it's forecasting from a date in the past into the present. And so what you see at the bottom is what that looks like. If you go to the CDC website, you see these different variants and the proportions over time and how they're changing. This methodology has remained mostly constant since 2020. It's a multinomial regression model in time. The secret sauce is that it accounts for heterogeneity in sampling practices across different states. But that's really that is the methodology that has been used for a while, and we've been sort of rolling around in our heads, how could we check the performance of this model and if we could really do better on top of it? And the pie in the sky dream is that you would be able to integrate multiple genomic signals to improve these forecasts. So right now that modeling just takes in clinical genomic data and then spits out the variant prevalence estimates. But really what you'd like to do is also incorporate things like wastewater data and maybe also other data streams. I think the CDC's traveler-based genomic surveillance system is really cool, and I'd also love to talk to anybody who's interested in that. This, of course, is a really challenging problem to do this data fusion because all of these different genomic signals are so different from one another. And so being able to account for the different observation processes and get the advantages of each system without their disadvantages is what's really important. So we have this hospital reporting system, just really nice because you get swabs, you get high quality individual genomes, but the turnaround is pretty slow. The wastewater system, you get this community-wide system, but then you have to deal with the complexities that come from the site-to-site variability and the bioinformatic complexity. There's this other system called traveler-based genomic surveillance, which is really cool. If you land at one of 7 airports in the United States, you can volunteer to give a nasal swab totally anonymously. And this actually, we did some work and showed that this predicted COVID prevalence in the UK pretty well, which I think is just so cool. Like, it's such a cheaper system that might be able to improve this kind of global monitoring. There, the turnaround can be really fast, but it's not the same population as all the other data that we're collecting, right? So figuring out how to incorporate all of those, I know that there's some value in there, and it's a matter for us to figure out how to get to all of it. While we're talking about complexities, no, I can't go forward anymore. Can someone take me to the next slide? Ooh, oh, that's exciting. The next slide, I'll talk, and then you'll see it on here, which is that an interesting thing about thinking about COVID-19 forecasting for COVID-19 variants is that the relevant entities to be modeled change over time. So the example that I'm going to give on the next slide. Ooh, can I do it? Nope. Maybe. Well, we'll check it. So if you think back to January 20, oh my gosh, 20. No. I can't remember when it was. It was in January of one of these years. When Omicron first hit the United States, you could say Omicron, and you might have had to say, oh yeah, I mean B1.1.529. But then, like a month later, when you said Omicron, you'd have to say, wait. Do you mean everything? Do you mean, or are you breaking out BA1? Are you breaking out BA2? So for modelers, the applicable modeling units, the taxa that are going to be in this model, are going to be driven to some degree by the cladistics, like by the actual phylogeny, but also by the epidemiology, because if you have 2 variants that are epidemiologically consistent, then they might be behaving the same way, and maybe you want to model those together. Or maybe the consumer of this data isn't interested in the complexities of the phylogeny. They just want to know, are we going to have problems with the variants that are going to need to change our clinical practice? And so that's what I think is also really interesting to think about if you're thinking about decision support. What are you trying to do with your model? I think for variant nowcasting, there's at least 2 goals. One is you want to know, am I going to have a wave? And then a second one is, when is this taxon going to achieve some level of prevalence? And they both have different importances. I mean, I think the most urgent question is, will this variant drive a wave? There's a huge number of downstream public health actions that follow from that. But my sort of touchstone for knowing that we haven't optimized that system is that there's a lot of discussion about will there be a wave? I'm not opposed to this discussion happening on Twitter, but I think the fact that on Twitter we are doing a lot of Bayesian integration in our brains means that we haven't optimized the models that we can do as much Bayesian integration out of our brains and in the mathematical model as we can. Another example that many people aren't familiar with, but was actually really important in the United States, at least, was that variant forecasting informed the deployment of medical countermeasures. So I have this example of, and I'm only going to say these names once, bamlanivimab and etesevimab. I practiced. So in February of 2021, the US FDA approved these 2 monoclonal antibodies. So if you're hospitalized, you can get these, and this will reduce your chances of even more severe outcomes. In the summer after that, distribution was paused because there was some fear that the beta and gamma variants, that this monoclonal antibody would be ineffective against those variants, so distribution was paused. It turns out that beta and gamma didn't spread very much, so we restarted the use of this monoclonal antibody. Distribution was paused when Hawaii had a certain amount of delta, because initially it was thought that this monoclonal antibody would be ineffective against delta. When we figured out that delta was fine, we restarted, and then when Omicron came around, this monoclonal antibody was kind of out, and now the emergency use authorization for it is revoked. So the point of all of this is that the people who were deciding where to move millions and millions of dollars of this drug around were really looking at the variant nowcast, and that was informing their their decision. So knowing what you're trying to predict actually really matters, because if a model that is good at saying, will this new taxon take off, could be a really different model from the model that says, given that this is the dominant taxon, when is it going to hit 50% prevalence, for example. So excited to have lunch, guys. We're almost there. And this is my last point, which is that there's likely utility in thinking not only about prevalence as a percent or as a count, but probably the two of them together. They each have strengths and weaknesses and are probably useful for different questions at different times. I think the dream, right, is that we would be building models that take in all the clinical data, take in all the sequencing, and really infer the clinical properties of new variants. I mean, I think that's a little bit also pie in the sky, but I think any model you need to ask which one of these two is helping you achieve your goals, not just whether it's accurate or not. And that's going to bring me to some conclusions and thoughts here, which is that my number one is to respect the data generating process. If there's heterogeneity in it, incorporate that into your model, even if it's statistical. Judge models based on performance rather than elegance. Like, really what should drive us is what works. On that line also, if you want your model to be useful to public health, it has to demonstrably outperform the methods actually used by public health practitioners. And so I think the wastewater community suffered a lot from this, because as a public health practitioner, you could just look at the hospitalization data that was coming in. You could tell if it was trending up or down. So if someone told you, oh, I have wastewater that also says the same thing, you as a public health practitioner would shrug and say, okay, what you're telling me is that your data didn't add any value to me. So I think that's why data integration is so important, and really emphasizing that your model can be effective by building it within an evaluative and comparative framework. If you can't prove that your model outperforms the existing method that is actually used by public health, even if that's eyeballing the data, then you're not going to get anywhere. And I think signal fusion is the future, but it's going to be hard. Okay, a lot of people were involved here in CDC, the Center for Forecasting and Outbreak Analytics, folks in the COVID division, folks in the genomics part of CDC, the forecasters in the forecasting division. This is if you're interested in. There's no write-up about it yet. It's coming, of the wastewater model in there. These opinions are all mine. And my final point, which is perhaps my most important, is that CFA is always looking for talent. So if you know anybody who's interested, even if it's tangentially or some years down the road, please either just talk to me or send me an email. I have the very transparent email, ulp7@cdc.gov. Thank you for your time and attention before lunch. I appreciate it.
Thank you so much. Any questions?
Just a quick question on one of your earlier points where you were talking about this, like, surging and the signal. And so are you comparing the signal to something that you would already expect to see? Because, like, thinking about when your denominator changes, like if you have Taylor Swift coming to town and everyone's coming, and then you're going to have a lot more people contributing to the wastewater system, how you disentangle an actual surge from your changing population.
Yes. I can't tell you. My job before this was at BioBot Analytics, a wastewater epidemiology company, and we got so many questions about, I'm a beach town, what's going to happen this weekend? I was like, oh, you know a lot more than I do. I think also a challenge with this kind of tool is that you have to train it on some data set, I think, is like the answer to that question, right? So you can only do as much with it as what your definition of surge is that you feed into it. And I think I don't think this tool has, I mean, I don't know. I know the authors, so I'm going to be careful what I say. Like, I don't think that this tool had super duper widespread utility, but I think it's a good example of the challenge of trying to do something with the wastewater data and look at these 3 or 4 data points that wiggled and then say, is this a big deal or not? And I think that's the strength of this tool, is that it actually answered the question rather than saying, well, wastewater data has high noise. But I totally agree with you. I don't think it tends to account for any of that.
Scott, great talk. We at the foundation work a lot with partners who, in those locations, even hospitalization data is largely syndromic and not like, you don't know what's causing disease. And so I'm just curious, with your wastewater informed forecasting, if there's a plan to test, if you were to integrate syndromic data with wastewater versus like case data with wastewater? Like what are the confidence intervals, and can we make some predictions based on that?
Oh, super. Yeah. Well, even the US system is changing from a census of admissions toward, it's like we're kind of flip-flopping back and forth, but there's going to be some syndromic surveillance will for a while be the primary signal, even for COVID. I think one of the things that's neat and that we're trying to do, which is also proving really difficult, I'm going to be putting my colleagues on the hook by saying this, is we're trying to build as like a modular system so that you can separate the epidemiological dynamics from the observation processes in the modeling. And so the ideal is that your observation modules you can plug in and pull out as needed, so that if you had a change in data stream, you could just pluck in or pluck out this module into your larger model. And so I think that's the dream too, is that you could use the same epidemiological backend, or you could have multiple epi models on the back end that you could plug in, that you could have different observation processes on the front end. And hopefully the answer isn't which one of these is best, but you create all the tools so that you can plug in whichever one is most appropriate to your epidemiological context. So maybe that's kind of a non-answer, but that's, I think, where we're hoping to go. And I think that's where the best place to end up is. Yeah.
Last question.
Thank you. Thank you for the presentation. I have a question about, like, you talk about like the model and how it works, but so how far have you tested it? And like, I mean, we have a lot of data available now from pandemic, and how is it doing? What's the accuracy of the models you've been developing?
Right. Yeah. And so I think this gets to this question about evaluative frameworks and so forth. And one of the things we really learned in developing that model was quite complicated, was to build the evaluative framework from the start. That one's being submitted to the COVID forecasting hub. So you can just like look at its performance in real time prospectively. There are ongoing work in that group to go back and say, if we had used this model as it is right now at every point in the past, and doing that evaluation. I think that the thing that I really wanted to see here is to compare, I can't point at anything, is to compare a hospital admissions only model to a hospital admissions plus wastewater model. So I think there's one question of how does this model perform against other models in an ensemble? But I think the thing that's really neat about this in a modular observation process framework is that you can just say, if I just remove the wastewater observations, to what degree does that degrade or improve my forecast, and when and by how much? And what we sort of see preliminarily is that the wastewater data is most helpful when you have changes like extrema in the time course, right? If your hospitalizations are going like this and they keep going like that, a hospitalizations only model is going to do really well. But if you hit the peak or a trough, then you start to get that reporting delay becoming really important. And those are places where the wastewater model can suddenly outperform the hospitalization only model pretty substantially. I don't have numbers off the top of my head, but they're working on it. Yeah. It's going to be a cool preprint when it comes out. Yeah.
Awesome. Thank you so much. Let's thank Scott and the rest of the presenters as well. This is an insane session.